All right, good afternoon. Let's go ahead and get started. Really no new announcements as usual. A new homework will be posted after class today. Keep doing them, keep handing them in. Most of you are handing them in for extra credit. Great, there's still plenty of more opportunities for you to do that. Even if you don't want to hand it in, I really highly recommend that you try to do the homework before you go to your discussion section. That's going to give you the best idea of how well you actually understand the material. It's really easy to fall into a false sense of security. If you haven't looked at it, you go to discussion, you hear the answers, be like, oh yeah, that's what I would have put. Oh yeah. Or you read the answer key, yep, that makes sense. No, you need to test yourself ahead of time. So make the most use of these homeworks that we're posting. Do the practice online exam. The pre-midterm will open next week after class on Thursday. It'll be open for 48 hours. By this point, who all has put on their calendar where they're going to take this? Good, a good amount of you. The rest, really schedule that time. Don't forget to do it. Then the IAs will hold a review session on Sunday in this classroom. We are aiming, it is scheduled to be podcast, but hopefully some of you can be here in person and go through the answers to the pre-midterm and make sure you understand the reasoning for the questions and why options are correct and why ones are not as correct. It's just as important to understand why something is not as correct, which is why we go through all of the options when we do the clicker questions. And then finally, on Tuesday the 28th, we will have the midterm in class. The pre-midterm and the midterm will cover everything up until the end of class next Thursday. Any questions? All right, so let's review what we talked about in class on Tuesday. So we started talking about this second major molecular mechanism, and that is the process of transcription. Transcription is using a DNA template to synthesize RNA. And for this, we are going to need the DNA sequence. This is going to serve as the template. It's what gets copied. And we had some conventions when we talked about the sequences around the promoter. And so by convention, transcription starts at the +1 position. Everything that gets transcribed after that +1 position is going to be moving in the downstream. The polymerase is moving down the template, and it's going to be positive. So +1, +2, all the way up until the end of that template that gets copied. Everything upstream or before the +1 sequence is going to be negative. In addition to the promoter element, which is going to be in the DNA sequence, we of course need proteins to do the work. And in general, we've been talking about RNA polymerase. And RNA polymerase is this complex of proteins. And in this class and subsequent classes, we'll start talking a little bit more about what is composed in that complex of proteins. So for transcription to happen, it first needs to initiate. Initiation is simply polymerase being placed on the promoter so that it can start transcription at that +1 site. After it uses the DNA template in order to know what the first nucleotide is and then the second nucleotide, and that first phosphodiester bond between those two nucleotides is synthesized, then moves on to the elongation phase. The elongation phase is just the polymerase moving down the template, using that template to know which nucleotide to put in next, until there is a signal to stop transcription. And as I mentioned, this is different in prokaryotes, it's different in eukaryotes. We'll talk about the main mechanism for terminating transcription of messenger RNAs in eukaryotes after the midterm. For now, just know that at some point, the polymerase is going to stop, it's going to detach from the template, and then you have your messenger RNA that can be translated into protein. All right, here's a clicker question for you. What are the first 4 nucleotides transcribed from the above DNA sequence? Pay attention to all the information I've given you up here. Ten more seconds. All right, final answers. All right, we have a lot of people voting for C, some votes for A, and then a few other scattered votes here. What is the correct answer? Oh, a lot of people like C. Why is it C? Explain your reasoning. Someone. Yes, Madeline. And that is what C is, right. So is the strand I'm showing you the template or the non-template? It is the template. It is what is being copied. So the key here is we always know we have to go in the positive direction. So if I indicate the positive direction, you know synthesis has to go in this direction. This is the 5' here. We have to have the 5' on the opposite side over here. And synthesis is always going to be in the 5' to 3' direction. Does everybody understand that? So why did I do that? Why did I put the pluses on this side instead of on the other side where we've always put it before? Was it just to confuse you? No, it's because genes actually on our chromosomes are in both orientations. If you look into the human genome, you will find genes that look the way we originally saw. So you have the +1 and goes to +10 in this way. So this RAB-EPK gene is being transcribed in this direction. So these blue boxes are actually the exons. While on the other side, way down here, is this HSPA5 gene, and it is being transcribed in this direction on the other strand. So a given strand of a chromosome is going to be a template or a non-template depending on the gene and which direction it's going to be transcribed in. Does that make sense? All right, so always pay attention to I either will have to tell you where the pluses and minuses are, where the 5' or 3' are, or tell you that it's a template or non-template. If you have any of those pieces of information, you will be able to write the RNA sequence that is being synthesized. Any questions about that? Yes, Lydia. So it all depends, and we'll talk later about this in class, where the promoter sequence is, right? So where the polymerase sits down, it will position it in the direction that transcription should go in. Other questions? Yes? Well, the sequence will always be the same. So it doesn't matter where it is located in the nucleus. The sequence is the same, and that's what these transcription factors we'll talk about more today are just looking for that sequence that it can bind to. Similar to when we talked about the Lac Operon, which was what we talked about when we talked about one example of how gene expression can be regulated in prokaryotes. And so we looked at this example of the Lac Operon. The whole point of synthesizing the genes from the Lac Operon is to use lactose to make glucose for energy for the cell. So the only reason you would want to turn on these genes is if you have the substrate available, right? Is if you have lactose available. If there is no lactose available in the cell, what's happening to this gene? Yeah, it's repressed. And how is it repressed? How is it repressed? So this complex of repressor protein binds to this sequence that's called the operator. When repressor is bound to the operator, it blocks the promoter polymerase can't bind. If polymerase can't bind, you're not going to get any synthesis. So if there's no lactose around, repressor is free to bind. Some lactose comes into your cell. What happens then? Yes? Yeah, exactly. So this sugar, lactose, actually binds to this protein complex, the Lac repressor. And when it binds to the Lac repressor, it changes its conformation, so it no longer can bind to the operator. Okay, that frees up this promoter sequence, and so now sigma 70 can recruit polymerase to this promoter sequence. But what's a problem with the promoter at the Lac Operon? Yes? It is a weak promoter. And why is it a weak promoter? What do we mean by it being a weak promoter? Yeah, go ahead. Keep going. Yeah, so sigma 70 has a sequence of nucleotides, the consensus that it's its optimal binding. It has the highest affinity for a specific sequence. If you change a few nucleotides in that sequence, it's going to have a lower affinity for it. And it turns out that the Lac Operon has a few nucleotide changes away from consensus. What that means is sigma 70 can still bind, and every now and then it'll bind long enough that polymerase can take off and start transcribing, but it's weak binding, and so you get a weak or low level of transcription. How can we increase the transcription? What state would we have the highest level of transcription? What else has to happen? So we have lactose. No glucose, exactly, because we want to metabolize that lactose so we get glucose. So when there's no glucose, there's a signaling pathway that makes a lot of what? CAMP. So it's adenosine monophosphate that has that cyclic group. So cyclic AMP. What does cyclic AMP do? Yes? Is it going to prevent CAP or? Yes, it's going to allow for it, exactly. So CAP, you can remember this C, this almost looks like CAMP, right? So CAP binds this little molecule CAMP, and only when it's bound to CAMP can CAP bind to this DNA sequence called the CAP site. The binding of this protein complex along with CAMP to the CAP site holds sigma 70. It helps stimulate sigma 70 and polymerase to be at the promoter and have high levels of transcription. So there's protein-protein interactions between CAP and this sigma 70 polymerase to hold it there so you can then get high levels of transcription in order to metabolize that lactose and make the glucose the cells need. Any questions about the Lac Operon system? All right, so today we're going to move on and start talking more about transcription in the eukaryotic cells. And first we'll do a transcriptional control overview, think about what it means to activate versus repress transcription, and then we'll talk specifically about the RNA polymerase, in particular RNA Polymerase II, and this motif it has, this C-terminal domain. All right, so when we have a gene that is wrapped up in tightly condensed chromatin, it's not going to be accessible to polymerase, and so that's usually going to be a gene that is in an off status. But if we want to activate or turn on expression of the gene that's wrapped up in this condensed chromatin, we're going to have a general set of proteins we'll call activators, some of which are going to be involved in changing this chromatin structure so that it can now be decondensed or loosened or opened up. What kind of, can you think of maybe one kind of activator that would help open up the chromatin? Yeah, if we acetylate certain positions on our histones, that's going to help open it up. So histone acetyl transferases would be a type of activator that might be recruited to that gene to loosen up the chromatin. As the chromatin gets unwound, there are then availability for other types of activator proteins to bind and eventually recruit this core RNA polymerase and all its accessory transcription factors so that the gene can be turned on. It can be active for transcription. Of course, gene expression is reversible. If we can activate gene expression, we can also repress gene expression. So this would be going in the opposite direction. So if we want to shut down expression of an open gene, there will be repressor proteins that will come in, they will block polymerase from binding, and they will start to fold that chromatin back up. What would be a type of repressor that might work on the histones? Yes? Yeah, methylation. So histone methyl transferases, methylation at certain positions, especially that lysine 9 of histone 3. It's going to package that chromatin back up. It's not available anymore for polymerase to see for transcription. So we'll talk more specifically about certain types of activators and repressors in the next few classes, but that's sort of the big picture of how we open and close genes to turn them on and turn them off. All right, so when we're talking about transcriptional regulation, it's going to involve promoters and transcription factors. What is a definition of a promoter? What is a promoter? What is it composed of? At the very simplest level. It's DNA, exactly. It is a DNA sequence. When I use that term element too, I'm always meaning a DNA sequence that regulates transcription. What are transcription factors? Proteins, exactly. So these are going to be these proteins that recognize these sequences and bind to the DNA and potentially recruit the polymerase to a particular gene for transcription to occur. So whenever I say promoter, I'm talking about a DNA sequence. When I'm talking about transcription factors, I'm talking about proteins that regulate transcription. And so it's very important to think about this because are promoters the same in every cell type? Is your DNA sequence the same in every cell type? Yes, the DNA sequence is the same, but how it is read, whether it can be turned on and whether you can actually activate transcription is of course going to depend on what transcription factors are produced in that particular cell type. So remember we saw like that globin gene in undifferentiated cells. It was in tight chromatin. There was no expression. But then as you differentiate those cells, certain transcriptional activators start to come on. They bind to the globin promoter and activate transcription. But all your cell types have the globin promoter. It's just only recognized for transcription in cells that are promoted to turn into red blood cells. Does that make sense? All right, so I mentioned before that RNA polymerase is actually a complex of proteins that come together to have this polymerase activity. When we were talking about prokaryotes, there are multiple subunits that make up the E. coli RNA polymerase, meaning beta prime is a separate protein from beta, is separate from alpha 1, alpha 2, and these just come together to form the active protein complex. In eukaryotes, we actually have 3 different RNA polymerase complexes called polymerase 1, polymerase 2, polymerase 3. Some of the subunits they have are quite similar, some of them are very different. Which one of these RNA polymerases is responsible for transcribing protein coding genes, making messenger RNAs? Do you know? It's the only one we're going to talk about from here until the midterm. Want to take a guess? 2. So RNA polymerase 2 is the one that synthesizes your messenger RNAs. And we'll talk about what Pol I and Pol III do after the midterm. So I showed you that one of these subunits has what's called, so Pol II, that's what we're going to talk about. It has this carboxyl terminal domain. Is the carboxy terminus at the beginning of the protein or at the end of the protein? It's at the end of the protein, right. So at the end of that subunit of RNA polymerase II, there is this sequence, these amino acids that are repeated in humans 52 times. So this group is repeated, so it's a 7 amino acid sequence repeat that's over and over and over again. Oh, these are single letter abbreviations for the amino acids. What is S? Serine. What about P? Proline. We have another serine. What about T? Threonine. Proline again, serine again. What about Y? Tyrosine. Very good. Okay, so you'll see why it's important to know what those amino acids are in a moment. So when RNA polymerase complex comes to the promoter and it's about to initiate transcription, it is in a largely unphosphorylated state. Okay. Can any of those amino acids that we just mentioned get phosphorylated? Which ones? Tyrosine, threonine, and serine. Exactly. They have hydroxyls that you're able to add a phosphate onto. And it turns out once the polymerase starts moving away from the +1 site, this carboxyl terminal domain, many of the amino acids undergo phosphorylation. That then allows that carboxy terminal domain to be recognized by other proteins that are going to be useful for helping the polymerase to transcribe, as well as to start processing the messenger RNA that's being made. So stars just indicate the amino acids that can be phosphorylated, which is really all of them except for the proline there. All right, so we said that we're going to need to have promoter sequence, the sequence that is going to be used to start transcription. This is the DNA sequence. There are 3 general promoter elements that we'll talk about here. For a gene to have any synthesis occur, for polymerase to be able to sit down, you need to have one of these present. Some genes are more complicated and have multiple versions. But we call these the core promoter elements. And you're going to need to have one of these present for polymerase to start transcription. So let's see how we define each one of these general promoter elements. The first one is called the TATA Box. And its job is to position the RNA polymerase at the +1 for transcription to occur. And it gets its name from the sequence that it has. So genes that have a TATA Box core promoter sequence, somewhere between -35 and -25, so counting backwards from the +1 in the negative direction, you're going to see this TATA rich sequence. That's why it's called the TATA Box. And then at the +1, it's usually an A, but other half of the time it's the other nucleotides. All right, so if I'm showing you the sequence going in the 5' to 3' direction here, and this is the +1, and this is a consensus sequence here. Are these sequences in the template or non-template strand? What's that? You said it right. I just didn't hear something dropped. So synthesis is going 5' to 3'. It's going in the same direction as the sequence that's shown. It's the non-template, exactly. So this would be the non-template sequence because 5' to 3' would be your template down here, and that's what would be copied in that direction. Does that make sense? So if I give you a sequence and tell you and ask you which core promoter element is there, if you look in the -25 to -35 region and there's a TATA rich sequence, it's going to be a TATA Box. The next type is called Initiator because it's based on nucleotides that are around the +1 site, but this is much less specific and much harder to identify. So an initiator core promoter is almost always going to have an A at the +1 position. And then at the -1 and -2 position, you could have a Y. Y is shorthand for a pyrimidine. Which nucleotides are your pyrimidines? C and T, exactly. So that means you could have a C or T here or a C or T here. The +2 position is N. N means it can be any nucleotide. The +3 position is going to be a T or an A. And then we're going to have pyrimidines again. So not a whole lot. You can imagine a lot of different combinations of nucleotides would give you an initiator sequence. So harder to identify. The third one is also often easy to identify, and it is called a CpG island. What that means is you have a run of C's and G's that are usually about 100 to 1,000 nucleotides upstream of the start site. So much further away than where our TATA box is, but you're just going to see a whole lot of C's and G's. That P in the middle just stands for the phosphodiester bond between the C's and G's. So if I give you a sequence and I ask you to identify which core promoter element is there, easiest thing to look for first is at -25 to -35. Do I have a TA rich sequence? It's going to be a TATA box. If you don't see that, and I show you a lot of nucleotides far up, and you see a whole lot of CGs, then it's going to be a CpG island. If you don't see either one of those, and there's an A at the +1, then it's going to be initiator. And I will never make it confusing that like, oh, maybe this isn't quite TA, okay? These are the 3 general core promoter elements. Polymerase needs to have one of those in order to sit down, find the +1 site, and start transcription. Now, one more thing about CpG islands. You'll notice that I have start site in quotations here. And that's because in contrast to the initiator or the TATA box, where transcription has a pretty set +1 site where transcription starts, for CpG islands, it's a little bit sloppy. You know, there's just a whole bunch of CpGs, so the polymerase gets positioned a little bit differently each time, and so sometimes it starts transcription here, sometimes here, and so there's a little bit of heterogeneity where the start site is. Is that going to change the sequence of the protein that is going to be translated from the messenger RNA that will be made from this? If just at the 5' end of that messenger RNA, you have a little bit of sloppiness, is that going to change the coding sequence? What do you think? What's that very first sequence that gets transcribed? Usually before you get to the start codon. The 5' untranslated region. So typically, this sloppy start might affect the 5' UTR a little bit, but usually the start codon will be much further downstream and it won't change the sequence of the protein that's made. The other weird thing about genes that have CpG islands is that you also get transcripts made in both directions. So every now and then, the polymerase will sit down on the other side and start transcribing in the opposite direction. It's still synthesizing 5' to 3', it just sat on the wrong face of that chromosome. These transcripts so what we call that when transcripts go in 2 different directions, divergent. So 2 different directions are divergent transcripts. The transcripts that go in the wrong direction that are not transcribing the gene, those are recognized as wrong transcripts. Usually transcription doesn't go very far and those are rapidly degraded. But that's what we mean by divergent transcription in genes that have CpG islands. We know this happens, but for the most part, it will not affect actual expression of the gene that you want to transcribe. Any questions about that? Okay, so now let's talk about regulatory sequences. So what we just talked about, that TATA box, the initiator, CpG islands, we call those core promoter elements. Your gene is going to need one of those, at least one of those, for polymerase to sit down and do any transcription. But it's going to get help from additional regulatory sequences to which activating proteins or repressing proteins might bind to that gene and either help polymerase activate it and transcribe or block polymerase from efficiently transcribing that gene. And so these sequences that regulate expression, regulate how those core promoter elements are read, are called either promoter proximal elements or enhancers. These sequences that are closer or more proximal to the +1 site are the ones that are called promoter proximal elements. So all these little orange boxes here, closer to +1, promoter proximal. Ones that are further away, we call enhancer sequences. And after we talk a little bit more about promoter proximal elements and enhancers, we'll then talk about how we would experimentally identify these types of control elements. All right, so let's talk about elements that regulate these promoters. So here's an example of a gene. At this, around this -30 region, we have indicated we have a TATA box. That is the core promoter element. And what's going to regulate how strongly that is recognized, how well polymerase is going to transcribe from there, are going to be these regulatory elements. Promoter proximal elements are defined as those that are closer to the promoter. They're more proximal to that +1 site. They're usually 6 to 12 base pairs long, a sequence bound by a specific DNA binding protein. Regulatory sequences that are much further away, usually 200 base pairs away, sometimes they are within the gene themselves. They can be in an intron sequence. They can actually be way downstream or way upstream, so 50,000, 100,000 nucleotides away, and yet be able to regulate how well the gene is going to be transcribed. These are called enhancers. And they are usually stretches of sequence that are a bit longer, 200 nucleotides long. So often it's a little fuzzy, the difference between these elements. You know, if you have one that's at 100 nucleotides away, is it an enhancer or a promoter? We don't really worry about it that much. It is a regulatory sequence that is going to be recognized by specific transcription factor regulatory proteins that will either activate expression or repress transcription. So how do we find such elements? You know, if we have this gene, there's thousands and thousands of nucleotides upstream of it, we can kind of pick out the core promoter elements looking at the sequences, but what about these regulatory elements? How do we find them? Well, one very powerful tool we use in the laboratory to find these regulatory sequences is by building reporter genes. So what a reporter gene simply is it takes advantage of an easily assayed gene product. So it is a protein that you can easily detect. And what you would do is you would take your promoter sequence of interest and you would put it in front of one of these easily assayed proteins, put it into the cell or the organism, and look for expression. So the point of building a reporter gene is to test the effect of a specific DNA sequence on transcription or expression. Some of the most commonly used reporter genes in the laboratory, you've probably heard of some of these, is beta-galactosidase. So you would have your promoter of interest driving expression of this protein coding gene. This, in fact, is the first gene in your Lac Operon. Normally what it does is if you give it lactose, it breaks lactose into galactose and glucose. But we have this chemical called XGAL, and if this substrate is seen by beta-galactosidase, it gets cleaved into galactose and this blue pigment. So you can visibly see this pigment. This only is blue when it gets cleaved. So it only can get cleaved if this reporter gene has been expressed. Luciferase. Where does luciferase come from? Anyone know? It takes this substrate, luciferin, and causes light emission, which there's kits you buy in the lab, and it tells you how much light was given off. You only get light if you have this enzyme. Nobody knows where luciferase came from? Yes? Flows. It is an animal that lights up. Fireflies, yes, exactly. So this is an enzyme that fireflies naturally make, and it's very important for their mating and all of that. But we can express this in cells, and if you give those cells luciferin, light will be emitted. That can be measured. It will tell you that your gene was turned on to a certain degree. Probably one of the most common ones, and probably everyone's heard of GFP. What does GFP stand for? Green fluorescent protein. Great name because that's exactly what it does. When this protein is synthesized and you shine UV light on your sample, you will see green. You will know that you got expression of this reporter gene. And the power of using GFP in all kinds of studies was recognized with the Nobel Prize in 2008. Where did we originally get GFP from? Where is this protein originally from? Jellyfish, exactly. So Shimomura is the one who isolated this green fluorescent protein from jellyfish. Martin Chelfie was the first one to make a reporter gene and put it into a live organism, in this case, C. elegans worms. And so he used a worm promoter sequence, fused it to GFP, and then all the cells where that promoter turned on glow green. And then our own Roger Chen, he was a professor here in the chemistry department at UCSD. He recognized, well, I know what the sequence and structure of green fluorescent protein is. If I just start making a few amino acid changes, I change the sequence, it turns out you can get that protein to not glow green, but all different colors of the rainbow by just making these slight changes. So now in the laboratory, we have access to the coding sequence for all these different fluorescent proteins. So if you wanted to put two reporters, so two different promoters, fuse and see where they're expressed in the same organism, you could fuse one to a red fluorescent protein, one to a green fluorescent protein, look at and see which cells are green, which cells are red, which ones are yellow, where they're both being expressed. All right, so here's more examples of how we can use this reporter strategy to find these regulatory elements. So here's an example. A student in my lab actually made this reporter gene. He was very interested in this gene he had discovered and wanted to ask the question in the C. elegans worms, where he discovered the gene, when and where during development of the worm is this gene being turned on? So all he did is he took the predicted promoter sequence of this gene. The promoter sequence can vary, but in standard practice, we use about 2,000 nucleotides upstream of the +1. You just fuse it to the coding sequence of GFP. We put it into the worms. They produce progeny that were then transgenic. And this is very exciting because we got GFP lighting up in what we consider the worm brain and all the neurons. So this told us that this promoter was driving cell-specific expression. It was only turning on in neuronal cells, so that's probably where that gene has a function. And so just to make sure you understand, when we put this gene into the organism, it's DNA, right? So the gene is DNA. If the promoter is read by the cell type, if the right transcription factors are there to turn on expression, you will get expression of GFP messenger RNA. And then the cell will take that messenger RNA, use the ribosomes to translate it into protein, and then that protein glows green when we shine UV light on the sample. Here's another example. So in this case, the scientists wanted to compare two different promoters, maybe two different genes. They wanted to know, are these genes expressed in neural stem cells? And they fused promoter 1 to LAC-Z, promoter 2 to LAC-Z, put those DNA sequences into these neural stem cells, and then added that XGAL chemical in there and looked, do I see a lot of blue, meaning that LAC-Z, this enzyme, was expressed to cleave XGAL into that blue pigment. And based on these results, which promoter would you say is more active in these neural cells? 2. You see way more blue here than you do up here. Would I get this same result if I put these same two promoters, let's say, into heart cells? Maybe, but maybe not. I could potentially get the opposite result, right? So there could be different transcription factors in the heart cells than in the neural cells. There definitely are different transcription factors being expressed in those two different cell types. And if this promoter 1 has the sequences that are recognized by those heart cell transcription factors, I'm going to then see high expression of this same promoter in the heart cells and maybe no expression of this promoter in that cell type. Question?
Does the tagging actually attach itself to the gene itself after it's expressed, so the expressed and product gene, or does it attach itself to the transcription factors?
So what we're making here is DNA sequence, right? And so we, in a future class, will talk about cloning, how we would actually fuse this promoter sequence to our reporter coding gene. And then you take that DNA sequence in the form of a plasmid. Many of you probably heard of a plasmid. If you haven't, it's okay. It's just a way to deliver that DNA into a cell type or even into an organism. Once that DNA is there and it's inside the nucleus of that cell, if the proper transcription factors are C sequences in this promoter and bind to them and they're activating, it will turn on, go back to this, expression. It will make the messenger RNA for that protein coding gene. That messenger RNA will go out to the cytoplasm. The translational machinery will make it into the protein, and then that cell has that protein. And in the case of GFP, that protein is just going to glow if you shine UV on the sample. In the case of our LAC-Z, we need to add a substrate because LAC-Z is an enzyme. So we just dump on our cells this XGAL. If the enzyme was made, XGAL will get cleaved. You'll see blue. If there was no LAC-Z made, XGAL will just sit there. It will not be blue. The substrate wasn't cleaved. And so that tells you that this promoter was not driving expression of your reporter gene. Does that answer your question? Good. Other questions about this? Okay, so when we were building these reporter genes, we're using a large sequence of DNA, about 2,000 nucleotides, so that we can see expression. But what we really want to do is to be able to determine the specific sequences, right? Every single one of those 2,000 nucleotides is not being bound by a transcription factor to drive expression. We want to find the regulatory sequences, the important activating or repressing elements. So how do we do that? One way we do that is we create a deletion series of our promoter sequence and then look and see how expression is affected. So for example, if you start off with this sequence of promoter sequence for this particular TTR gene, what you would do in your deletion series is you'd start with your full, let's say, 2,000 nucleotides, and then you would start making it shorter, 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 shorter. And you would take these shorter fragments of promoter and you would clone it into a vector that already has your reporter gene sequence. So let's say this is GFP right here. So you'd put the full-length promoter driving GFP mRNA expression this way, a little bit shorter, a little bit shorter, a little bit shorter, a little bit shorter. And then you take each one of these clones separately, put clone 1 into a set of cells, look at expression, clone 2 into a set of cells, look at expression, clone 3, and so forth. And then you would have some sort of readout, maybe how much GFP are you detecting. If you see a lot of GFP, that means that this segment of promoter sequence is driving high levels of transcription. All right, let's break this down and look at each step in a little more detail. So the first thing we said we needed to do was to make these reporters. So we needed to have this promoter sequence, so this upstream region of the gene. Often you will include a little bit of the downstream sequence as well, because as we saw, for example, our initiator core promoter, it includes some sequence like beyond the +1 site. So you often include some of the sequence here. And how do you think, what method or what technique would we use to get this specific sequence of DNA that we want to use to clone into this reporter? We could use a restriction enzyme. When we're cloning, we need a lot of this DNA. PCR, exactly. So all you would need to do is design primers to amplify this entire sequence of DNA. And so you would therefore have the specific sequence that you want. And then look what happens. So if we want to get shorter and shorter and shorter to be put into our clones, you can use the same primer. Sorry, this is not drawn right here, but you would use the same primer on this side, and then you would just move your primer in to make shorter, shorter, shorter little fragments. You always want to move in from the position that's furthest from the +1 site. What would happen if you started shortening from this end? What would you lose immediately? The core promoter element, right. So you want to move from the furthest away to find sort of the shortest fragment that still gives you full activity. And then once it doesn't, how are we losing activity? And therefore, we might be removing regulatory elements. So this, we would use PCR to make what we call our 5' deletion series. And then we would use recombinant DNA techniques like cloning. We'll talk more about that after the midterm, but for now, just know that you could take your PCR fragments and clone them or insert them right before your reporter gene. The reporter gene, let's just say it's GFP, which simply means this encodes a messenger RNA that would be translated into GFP. And so you would make, in this case, 5 different mutants, so 5 different clones that only differ by how much promoter sequence you have included. You then take each one of those deletion mutants and you put it into your cell system, your expression system. And so once that DNA gets into the cell, gets into the nucleus, it can potentially be recognized by the transcription factors in that cell to recruit polymerase to your reporter gene, turn on transcription, make the messenger RNA goes out to the cytoplasm, makes the protein, which could be GFP or could be a reporter enzyme like luciferase, whatever you've designed to be your easily read out reporter gene. And then you just analyze the results of each set of cells that had one of your different reporters in it to look at where expression changes. So our first plasmid, let's say this was just the full length, the entire promoter sequence we wanted to analyze. So when we have all that sequence there, we get a reporter expression level. 3+ is higher, so highest level of expression. When we chop off this sequence, what has happened to expression? Nothing. It's exactly the same. So there is no regulatory element in this part of our promoter sequence. Does that make sense? Okay, we chop off a little bit more. Ooh, what's happened now? We have lost a regulatory element. Is it a positive or negative regulatory element? Positive, exactly, because we needed it to have higher expression. When we chopped it off, we got less expression. So we removed a positive regulatory element. All right, what about when we go to 3 to 4? Have we lost anything else? Nope, nothing's changed. What about 4 to 5? We've lost another element. What do you think we've lost if we get no expression? It's positive, and it would probably be one of your core promoter elements, right? If you remove a core promoter element, you're going to get no expression at all. So when you get these types of assays, all you need to look for is, where do I see changes in expression? That means that somewhere within this position, there was a positive regulatory element, some piece of DNA sequence that a transcription factor must recognize in my cells to promote high levels of transcription. Again, when we remove this sequence, we lost all expression, so I probably got rid of something that's really essential, like a core promoter element. Any questions about this? All right, let's do a clicker question. So, lots of data up here. So which of the following statements are supported by the data? So what you have here are 5 different reporters where we have done this same kind of deletion analysis. And we've put these reporters either into heart cells or into brain cells. And then read out expression. More pluses means more expression. 10 more seconds. All right, final answers. Ah, yeah, E seems like a pretty safe answer here, right? Yeah, just got to be more than one of the above. Let's see if that is accurate. So let's go through each one of these and make sure we understand how to interpret these types of data. So A said an element that activates transcription in heart cells is missing in reporters 3 through 5. Who thinks that's correct and wants to explain why? Or why not? Yes?
As we see like 3 through 5 compared to genes 1 through 2, there's clearly like a lot less expression. And since we can have multiple different transcription factors or something that activates transcription, there's probably something that's missing in reporters 3 through 5, causing the expression to be much less.
Yeah.
Not expressed at all in 5.
Yeah, exactly. So the question is saying an element that activates transcription in heart cells is missing in reporters 3 through 5. So the first thing I see here is like, oh, I only have to focus on heart cells for this question. And I want to look at reporters 3 through 5 and compare to the full-length reporter, what we started with. Yeah, we have definitely lost something that was activating. So by removing some sequence in here, right, we have lost expression. We have lost an activator. Is that clear to everyone? All right, what about B? B says reporter 5 contains an element that represses transcription in heart and brain cells. So that means we're going to compare reporter 5 to the original one that had no deletion. So does this have a repressive element in it? Is B correct? So reporter 5 has this sequence, this element. Is that present in every single one of the other reporters? Yes. So does it have an element that represses transcription? No. When it's here in our full-length reporter, we have very high levels of transcription. What do you think we've lost? So you can see that when we go from our reporter 4, we have some expression, and then whether we're in heart or brain cells, we lose all expression. What have we lost? Core promoter. And core promoter is an activating sequence, right? It's a way to get any kind of activity. So we certainly did not lose, or it certainly does not have an element that represses transcription. Does that make sense? Okay. So B is not correct. Let's see if any other ones are correct. C says the full-length promoter is more active in heart than in brain cells. Is that one correct? I'm seeing people say yes. Why? Yes. Absolutely. So I'm telling you the full-length promoter, that means the promoter before you started deleting, all you have to do is compare in heart versus brain. We have high activity in heart and we get no activity in brain. So it is definitely more active in heart than it is in brain. Same DNA sequence, but you're getting different effects in different cell types because there are different transcription factors in the different cell types. So C is correct. Does everyone see that? What about D? The promoter contains both activating and repressive elements that function differently in heart versus brain cells. Is that true? Are there activating sequences? Okay, where are they and when are they activating? So we said before that we lost activating sequences when we chopped off this sequence, right? Because in heart cells, we had high levels of expression and then they went low. So in heart, this sequence was recognized by a protein that is activating transcription. With that same idea, what is happening in the brain cells? No expression. So is there a repressive sequence there? So that means when this sequence is present here, there is some kind of transcription factor, some kind of repressor factor in brain cells that's sitting down on it and blocking transcription. So it is the same sequence, but it is being read differently in the different cell types. What will determine if a sequence is activating or repressing are the protein factors that are going to recognize it and sit down on it. Does that make sense? All right, so E was correct, and in fact, all of them are correct except for B. I just want to make sure that we are clear about this activating versus repressing, and so I've sort of drawn out how you could visualize this. So if we're just looking at the heart cells where we have high levels of expression, you can imagine that there is some activating protein that is only present in heart cells that needs this sequence to bind to, right? Because when we remove this part of the sequence, we still got activating, so we know it's not out here, but it's got to be in some region that then gets removed with our mutant 3. And what that activating sequence does is maybe it holds polymerase down to the promoter better, so you get higher levels of transcription. Then, when you remove that sequence, when you've gone down here, you still can have polymerase bind. There's still core promoter sequences that allow it to come in until they are lost in your last one here. Then you no longer get polymerase bound and you no longer get expression. So that's how you could imagine there being an activating protein present in the heart that recognizes this sequence, activates transcription. Question?
I just want to be clear. So we know there's a repressive element in the heart because of what we see in the brain. It's the same sequence.
Right, so it's only going to act as a repressor or an activator if the proteins are there to read it. If this heart protein, that is the activating protein, isn't there in the brain, but there's a different protein, different specific protein in the brain that will bind in this same region, maybe the same sequence, but maybe some other sequence. You know, we're probably deleting hundreds of nucleotides here. And it sits down, it may block RNA polymerase from binding, similar to how in the Lac Operon, Lac Repressor can block polymerase from binding, and then you get no expression. And so as long as that sequence is there, which it is in reporters 1 and 2, you get no expression. You get rid of it by deleting that sequence. Now polymerase can bind, this repressor isn't blocking it. And it just so happens there's no other activating sequences in the brain, and so you get low levels of expression until, again, you lose the core promoter sequence. Does that make sense? And these slides will be in the clicker questions tonight. All right. So these are all examples of how we would do deletion analysis. Simply march down chopping off sequences to see when we get changes in expression. But there's another way that we can more finely map these sequences that are important for regulation. And that is by using mutagenesis, so changing the nucleotide sequence instead of just chopping off sequences altogether. So this is often what we would do. So maybe if we knew nothing about a promoter region, we'd probably start with deletion analysis. So take those 2,000 nucleotides, start chopping down until we see some changes. Okay, so now we know we can get all the way down to 1,000 with no changes, but now changes are going to start. Then you might use that 1,000 nucleotide promoter to do mutagenesis systematically along that sequence to identify the specific regions that are regulatory. So once again, you would create a series of reporter genes, but instead of chopping off sequence, you would mutagenize a region, and then in another reporter, you'd mutagenize another region, and so forth. And then test each one of those reporters in your expression system, and then read out the reporter expression. So here's an example of what that could look like. So this just says expression level. So we start out here with our control region. Let's say it's driving expression of GFP. If we have this whole region here, the highest level of expression we can get is 3, so high expression. And then what you would do is you systematically start mutating sequences. So in mutant number 1, these dashed lines indicate that we've taken whatever sequence was here and scrambled it. Does that matter for expression? Nope. So no regulatory sequence within this region. Mutant number 2, we've now scrambled this region, and often we will overlap regions that we scramble because that can give you important information. What do we see with mutant number 2? Expression went down. So have we messed with an activating sequence or a repressing sequence? Activating, right. Expression went down. The activator can't read this anymore. Okay, but we can narrow down which sequence it is a little bit more because we already know this sequence that was scrambled up here didn't matter, so we know that sequence doesn't contain the activating part. Maybe it's within this region, and we can verify that with our mutant number 3, where this whole region was mutated, this part overlaps, and aha, we still have this reduced level of expression. So the overlapping sequence where we saw the change in expression must have an activating sequence. Now we go down to number 4 here, where we've mutated this sequence. Anything important in there? Nope, no expression change. Go down to our next mutant. Aha, what has happened here? We've lost activating or repressing sequence. Another activating sequence, right? And we can narrow it down to this small region because the mutant 4 and mutant 6, which has overlapping regions, doesn't matter if you change those sequences. So only this little fragment of sequence here that didn't overlap must contain that activating sequence. And then you keep going, no change. Ooh, we lost all expression when this was mutated. What did we lose? The core promoter, which is an activating sequence. Then you can move beyond and you can mutate this sequence and you get back to exactly what you were with the control. And so by doing this, you're able to map regulatory sequences within a promoter region. And so if you look at the transcriptional control region, it actually, for this particular gene, this turned out to be a TATA box, and then these are 2 promoter proximal elements that were important for full expression from this transcriptional control region. Any questions about that? Yes?
So it's kind of like opposite of what you would think. So like an activating sequence is going from like 3 positive to like 1, and then the activating is from like 2 to 3.
I don't like to think of it as opposite of what you think. So if you think about it, if you have something that is needed for an activator to sit down and you lose that, then your expression is going to be lower, right? But if you have a sequence that a repressor is going to sit on and you mutate that, pressor can't bind to it anymore, your expression level would go up. Would it go up higher than what you have in the original unmutated one? You've removed a repressive sequence, or you've scrambled, mutated a repressive sequence. Would expression be higher? Yes, it would be higher. You'd have 5 pluses, 6 pluses, whatever. You've taken away a repressive sequence. Does that make sense? All right. Final thing I want you to keep in mind is maybe you're thinking, whoa, these enhancer sequences, they're so far away. How can they possibly affect expression of a gene by helping recruit polymerase around the +1 sequence? And here is where I want you to come back and remember that the DNA, the chromatin, actually can fold back on itself. There's these scaffolds that hold different parts of the chromatin that is looped out. And what that means is very distant sequences in 2 dimensions can be very close together in 3 dimensions. So an activator that maybe is even downstream of gene B, so after transcription would actually start, could be very close to the transcriptional start site of gene A, and the proteins that are bound here could contact activating proteins here and help recruit polymerase to that +1 site in order to get transcription. So you have to think sort of in 3 dimensions for how these distant sequences can be brought into the proximity of transcription. And this is why this reporter method does not always work, especially in our complicated mammalian genome. There can be regulatory elements that are hundreds of thousands of nucleotides away, and so it's often difficult to identify all of the regulatory elements by using this method. But you definitely will find some of the very important ones. Any final questions? All right, have a great weekend.